Good morning, everyone. I'm Kyle, an assistant professor at Stanford. I'm actually a stem cell and developmental biologist, so you might be wondering, why am I here? It's because in the past few years, our lab has begun working on BSL-4 viruses, and it seems that after this, no one is rotating in our lab, which is a bit of a problem. And I'm really excited to be at this. It's actually my first virology event. I really want to thank the organizers for welcoming me into the Bay Area virology community, and for the last speaker, who's here. RTP4 is actually one of the top most induced genes in our endothelial cells after infected by Andes virus, the hantavirus. So when you mentioned that, I've read your paper, it's like, oh my God, like someone's talking about this. So maybe we need to talk together later. We don't work on flaviviruses. So all of you know that BSL-4 viruses are among the deadliest viruses on Earth. And one common feature is that they tend to cause vascular disease. So today, I'll focus on two viruses, Ebola virus, which needs no introduction, and Nipah virus, which has a 57% fatality rate. So one of the key challenges in the field, we think, is creating healthy cell types to study BSL-4 virology. Currently, the workhorses of BSL-4 virology are cancer cells. As all of us know, these offer great advantages, but they have several shortcomings. They're genetically abnormal. They sometimes lack the ability to secrete interferon, which you just heard about in the past talk is really critical to study viral cell biology, and they often incompletely resemble native cells. And so all of you know that human pluripotent stem cells, or HPSCs, offer the ability to create healthy cells. In cell biology, it's really been now increasingly accepted that HPSC-derived cell types really provide a powerful platform to study normal and disease cell biology. So our lab has developed methods to differentiate HPSCs into over 24 different kinds of cells. And today, I'm going to focus on the mesoderm germ layer in red, which gives rise to the vasculature. So first, we developed methods to convert human pluripotent stem cells into the two kinds of blood vessels, artery and vein endothelial cells. We've then used these human vascular cells to study the effects of these deadly BSL-4 viruses. In the first part, we found a surprising twist that Nipah virus did not infect all blood vessel cells equally, but actually preferentially attacked artery cells. And to our knowledge, this was maybe the first discovery that there's an important difference in arteries versus veins in viral infection. So this is a photo of the BSL-4 lab. My lab members and I have begun training to work there, but I really want to thank my stellar collaborator, Joe Prescott, who the CZ Biohub hosted last summer to give a talk. And he's really been my foray into this interesting world of virology. So I have to say, undergoing the training is an interesting experience. You have to get a security clearance and also a background check. So then you have to fill out all your social media accounts. So my lab members told me, like my grad student, like, don't look at my Instagram. And I told them I don't really know what Instagram is. I really don't want to look at your Instagram. But the reason, of course, is they're looking for insider threats. And on your Instagram, if you write something against the federal government or something, they're going to find that. So you have to be very, very careful what you say publicly nowadays. One key question we had were the effects of BSL-4 viruses on blood vessels, in particular the two kinds, arteries versus veins. So first, we differentiated human pluripotent stem cells into over 90% pure batches of artery and vein cells. And this occurred in just 3 to 4 days. So for those of you who are used to working with stem cells, this is very rapid and very efficient differentiation. And the way we did this is that we discovered at each step of differentiation what are the combinations and timings of developmental signals that we need to turn on and off at each stage. So for instance, one of our discoveries was that TGF-beta in red induced arteries, and another signal, PI3K in purple, induced veins. So we control these signals to sharply guide cells down one path or the other. To track differentiation, we have to use markers. So a transcription factor called SOC17 is an artery marker, and NR2F2 is a vein marker. So just keep this in the back of your mind, because those come up in the next 2 slides. I also want to mention that this work was done together with Lei-Tang Ang, who's now an assistant professor at Stanford. So how do we induce artery cells? We thought, OK, maybe we should activate the artery-inducing signal TGF-beta. But of equal importance, we have to block the vein-inducing signal PI3K. And this worked remarkably well. So in just 3 days of differentiation, we can now create over 95% pure batches of artery cells. So in the y-axis, you can see that these endothelial cells express the pan-endothelial marker, VE-cadherin, or vascular endothelial cadherin. And then the x-axis, they express SOC17, that key artery-specific transcription factor I mentioned previously. So I just want to emphasize on this slide, what's really key to our approaches is that we're activating one signal and inhibiting another to sharply guide stem cells to your desired path and to block unwanted differentiation. So we thought, OK, if we can make arteries, can we now make veins? So we just applied the converse signals. Now we activate PI3K and we inhibit TGF-beta. We can now make an over 85% pure vein population that expresses VE cadherin, but now the vein-specific transcription factor NR2F2. So for the aficionados in the audience, you'll know that in development, it's not just controlled by 2 signals. I'm just showing these for the sake of concision. The full list of signals can be found in our paper. So what do these endothelial cells look like before we begin using them for virology? So you can see the artery cells form these beautiful matted vascular networks with cyan VE cadherin. And in the nucleus, they express that transcription factor SOC17. What's pretty remarkable is that if you look in the DAPI channel for all the nuclei, you can see that virtually almost every single cell has been converted to an artery, emphasizing the purity of our differentiation. And this has now been reproduced by over 12 labs worldwide. Conversely, the vein cells, you can see they form these beautiful vascular networks of VE cadherin, but now in the nucleus, they express NR2F2 in green. And you can really see the high purity of arteries or veins that we produce. So having made these really pure batches of artery and vein cells, we then use them to study multiple BSL-4 viruses. And first, I'll focus on Nipah virus. So Nipah virus is probably familiar to many of you, but when I speak to a general audience, I have to remind them that it has a 59% fatality rate, which is pretty shocking. There's currently no FDA-approved treatments or vaccines. It's the only BSL-4 virus that's been shown epidemiologically to be spread airborne between humans. There's almost annual outbreaks in Asia. So it first emerged in Malaysia and Singapore, where I had a great discussion with Ui back in the audience. And now there's almost annual outbreaks in Bangladesh and India. And it enters cells through receptor protein on the cell surface called FNB2. So just keep FNB2 in the back of your mind for the next few slides. So while this is familiar to many of you, when I talk to a general audience, I have to tell people that Nipah virus is the virus from contagion. And you might think, OK, is this really the case? And it is. So I've looked through every single second of Contagion on Amazon Prime Video. And there's actually multiple great parts to this story. So they even have the crystal structure of Nipah virus envelope protein engaging FNB2, and with the contact residues highlighted. And if you look into it, it's a Nature Structural Molecular Biology paper with that exact same figure. But in this figure that I found, you can see they're using a DNA homology software. You can see that Nipah virus is a complete match to this green virus in the movie. And you might ask, OK, what's this number next to Nipah virus? This is the actual accession number for Nipah virus. And we actually align our RNA-seq reads to this genome and design an in situ probe. So I tell people now there's a lot of despair that Joe said that people don't want to go to biomedical research. But in Hollywood, they employ lots of PhDs now to fact check all of these movies. So there's a lot of job opportunities, at least I tell my lab. So the real reason that we got into Nipah virus is because of this FNB2. So FNB2 to us is really special in the vascular field because it's actually known to be a marker of arteries. And you can see in our cells, FNB2 protein is at 10 times higher levels in arteries than veins. Thought, that's really interesting. So while we would assume that viruses would infect the entire vasculature equally, maybe Nipah virus preferentially attacks artery cells. And so we found that this is the case in our lab with the BSL-2 pseudovirus, but the real proof came from our collaborator, Joe Prescott. In the BSL-4, he found that Nipah virus replicated to 11 times higher levels in arteries in red as opposed to veins in purple. And I hope that you appreciate this really stark order of magnitude difference. And to our knowledge, this is the first demonstration of an artery-biased virus. And I think also illustrates the importance of creating physiologically relevant cell types to study these viruses, because you wouldn't discover this with generic endothelial cells or cancer cells. So what are the effects of Nipah virus on these cells? So it's well known that Nipah, like other paramyxoviruses, induces cell fusion. So what's really striking to us is that if you infect vein cells with Nipah virus at a pretty high titer, after 24 hours, they look pretty happy and healthy, as you can see here from these zoom-in images. But Nipah virus completely devours artery cells. So in just 24 hours, the cell culture becomes this weird web that begins peeling back from the plastic of the plate. And what you can see is that up to 23 nuclei are in a single cell, indicating the fusion of many, many, many cells together. But what's also really interesting to us is that in a healthy syncytia cell, like a skeletal muscle cell, the nuclei are actually spaced apart, if you ever look at photos of skeletal muscle. But here, in these Nipah-induced artery syncytia, all the nuclei draw together like a bunch of grapes without fusing together, but kind of coalescing in the middle of the cell. So we wanted to prove that this was actually a cell fusion. So we designed an experiment where we created both a wild-type and GFP-expressing artery cells in a co-culture. And then in the BSL-4, these were infected by Nipah virus to see whether they would fuse together. So this is actually a technically very difficult experiment, and actually the second time in history, to our knowledge, that live imaging was done in the BSL-4. And the reason can be found in the song "A Hotel California" by the Eagles, that you can always check in, but you can never check out. So when we took a scope into the BSL-4, of course, no one's going to come service it anymore. So this thing lives permanently in the BSL-4. So in this movie I'm about to show you, it spans 8 hours. And before I press play, just want you to note one thing, that this is a co-culture of GFP-positive cells in white and wild-type cells in black. So when I press play, 2 things are going to happen. First, you'll see cells fusing, so that's white GFP moving into formerly black spaces. But you'll also see something really weird, that the nuclei have a life of their own, and they begin this kind of horrific aggregation. So you can now see these new cells appearing like they're coming from hyperspace, indicating fusion. But you can see that in these very, very large syncytia, the nuclei all begin coalescing. At this point, there's almost no black spaces because the entire plate has fused together. So for the people that watch Contagion, I tell people this is what happens in Kate Winslet's arteries. Of course, she doesn't make it to the end of the movie. So in the last few minutes, I just close on the cell biological effects of what Nipah does to arteries. So we infected arteries with Nipah, or as a control Sendai virus. And as you know from the previous talk, when many viruses infect human cells, they trigger interferon, which rallies antiviral defenses. So what we found is that in that entire movie of Nipah infecting, replicating, and fusing these cells, there was essentially no interferon response, which was really staggering to us. So in this heat map, you can see that we add Sendai virus, which is non-pathogenic. Although Nipah virus occupies less than 1% of the transcriptome, there's a massive interferon response, and all the IHGs you heard about turn on. But what's really staggering is that for Nipah virus, although it occupies 17% of the cell's transcriptome, so 1 out of 6 mRNAs are viral, there's really no interferon production. So it's well known that viruses inhibit interferon production, but this is to a really surprising extent. So you might think I maybe just cherry-pick those genes to show you, but it's really not the case. So when we infect cells with Sendai virus, there's very huge up and down regulation of many genes. But when we infect the cells with Nipah virus, after 10 hours, there's really 0 genes across the whole transcriptome that turn on and off. So the degree of innate immune suppression is really profound. So in summary, we've created over 90% pure artery or vein cells in 3 to 4 days of differentiation. We've used to find that Nipah has this predilection for arteries and causes them to fuse together, but is incredibly stealthy in that whole process of fusing and replication doesn't really change gene expression. So in the last minute or two, I'll just close on one piece of unpublished data that now that we've discovered the effects of Nipah virus, we're now taking on the challenge of the symposium, which is the comparative virology of how does it stack up to other BSL-4 viruses like Andes or Ebola virus. So this is a collaboration with Isabel, a postdoc in Joe's lab, my grad student Anna, and my collaborator Lei-Teng Ang. So although all these viruses are classified in BSL-4, they have and cause vascular diseases, they're very different. So Andes is a hantavirus, Nipah is a paramyxovirus, and Ebola is a filovirus, of course. So as you know, when most viruses, like Sendai virus, infect cells, they induce what we call a balanced immune response, turning on both interferon and inflammatory cytokines like IL-6. But we found in this comparison that Ebola is quite the outlier, and while it occupied over 50% of an artery cell's transcriptome, there was almost 0 interferon production. You can see on the y-axis is log 10-fold, so there's really no interferon induction. But the reason that Ebola was an outlier was that you can see in the gold star, it induced very, very high levels of IL-6. So we think that Ebola has this really interesting effect where it may induce what we think naively is the perfect storm, that Sendai virus induces both interferon and IL-6, but Ebola virus leads to this extremely imbalanced response, was extremely stealthy. It occupies over half of the transcriptome, but does not turn on interferon secretion, but now massively turns on IL-6. And this may explain 2 clinical features of Ebola disease. A, the lack of interferon production could lead to this really unchecked viral replication. Indeed, in Ebola, there could be over 2 billion Ebola genomes per ml of patient blood, which is pretty remarkable. But the induction of IL-6 could lead to this really strong cytokine storm. Indeed, in patients, it's known that IL-6 levels can actually be double that found in CAR-T-induced cytokine storm. So of course, there's many questions. How does Nipah inhibit interferon production? And can we use this knowledge to make viral gene therapies stealthier? how does Ebola have this really interesting effect where it can now decouple the two arms of innate immunity by powerfully blocking interferon production, but really dialing up IL-6, which is a really, really unnatural response. And now this provides a platform for comparative virology of how do all these other BSL-4 viruses infect the human vasculature. We're really excited to discuss ideas and collaboration. So in closing, I just want to thank my great collaborators, including my grad student Anna, our lab members, and support from various sources. And a final plug, if anyone here works on SADS, which is this alpha virus that infects swine, we'd really love to talk with you.
Great job.